Okay, so thanks everyone for joining us for today's session. It's probably the last we're going to run this year in the series, which has been fairly well attended, although unfortunately we don't seem to have hooked in any new projects, Matthew. We haven't had a very good response from the new projects. I'm not quite sure why.
Yeah, we'll have to do some recruitment on that front. Sorry.
No, it's fine. Maybe they're all, I did get a couple of emails from people just saying they're still finding their feet and, getting things going. So they were, but not quite at this, I guess, at the place we're at yet. But anyhow, okay, so today this is session 7 of the 2022 seminar series. And your hosts are myself and Jeremy Shelton. We're from the Freshwater Research Centre, and we've really been coordinating these series of seminars for making, hopefully at the end of the day, life easier for those who follow on from us and to learn from each other and develop networks and collaboration and hopefully create some nice synergies between the different individuals, organizations, and projects. So for today's session, we are going to be focusing on the taxon master lists, or how you create master lists for checklists, master lists, how you deal with taxonomic backbones, etc. And what Jeremy and I did in sort of preparation for these various focused question and answer sessions is to kind of delve into the previous year's seminars and pick out some of the topics that either had come up in conversation or in the discussion and that we thought would benefit from sort of more in-depth discussion. And we've had 6 of these this year, all of which are recorded and available for anyone who wants to view them at any stage. But today's topic is all about freshwater taxon lists, and we've sort of just jotted down a few questions. These are by no means exhaustive, and that's obviously just to kind of guide the conversation today. But the idea is to get input from anyone who's in the Zoom room. So the questions to pose are like, where are the different projects getting their taxon lists, and how do they, what resources are they using within their country to generate these lists? Have any projects had any issues with using GBIF as the taxonomic backbone? In the FBIS system, we made a decision early on to use GBIF as a taxonomic backbone. And I am aware of a few issues, but it'd be really good to see what other projects have experienced and sort of get some general input there. And then have any projects used the Freshwater Animal Diversity Assessment or the FADA lists? And I'm really grateful that Astrid's in the room, because she can probably enlighten us a little bit more around these lists. They did provide, she gave a presentation in the first 2021 series about the FADA lists, as well as a few other aspects that were very interesting to the group as a whole. And then lastly, how do we obtain lists and how do we keep these lists accurate going forward? And I'm sure that everyone will have some input on this. So I'm happy to sort of either just open up for general discussion, or let's dive into the first question initially to see, of those of us in the room, who is involved in generating lists and what have you used up until now. So just feel free to raise your hand and then we can just start the discussion. That's perfect.
Yeah, maybe I can start right now. But I think in Europe we do have another approach, because when we built up the data portal on the Freshwater Information Platform, we wanted to build it for the whole of Europe. So we were not looking for national checklists, but rather for entire lists for the whole of Europe or the globe. And that is why we actually used the FADA lists, and also because the Belgians, who are the producers or the editors of these FADA lists, they were also involved in our project where we set up the Freshwater Information Platform. So this was kind of logically to use those lists. And they were readily available at that time because they were produced in 2006, and yeah.
Okay, and I know that just from our speaking independently, though that project is still ongoing, although I think the web interface is sometimes a little tricky to use because of lack of funding and what have you.
No, you mean a web interface of FADA? So, so the project was mainly in around 2006 or something, and at the end of the project, they had this hydrobiologica publication, and they also put all the lists on their website. And then we jumped in with the BioFresh project, which was from 2010 to 2014 or something. And during this time, we could still maintain this FADA website as well. But after that, the funding got complicated, and then the main person who was responsible for this, Eike de Wever, he left the institute. So since then, these pages are not updated anymore, and that's why, yeah, that's why you can account difficulties with them. But as I said to you already when we were talking independently, we are trying to get funding for updating the whole of the FADA lists. That means we are applying for funds that we can revive the consortium that is behind the FADA list. So FADA consists, for those who do not know, FADA consists of mainly all animal taxon groups that occur in freshwaters, and each of these organism groups had a dedicated consortium of taxonomic experts who took care of establishing the lists. And now, after I think it's already 16 years or something, wow. We want to revive that. So that means we want to contact the different consortia of the different organism groups again, and then ask them to update the lists. And this would also then include that we update the website, and we are also working on having these new taxa lists then as taxonomic backbone for GBIF as well. So this is the plan, but yeah, the proposal is due in mid-December, and we will know, I don't know, somewhere in January if it gets funded or not. I very much hope that we get the money for it, and I have no idea how to proceed if we don't get the money. But for now, let's hope. Yeah,
no, definitely. So Astrid, if I can ask you though, so obviously the focus was Europe, but to what extent?
No, for the FADA list, it was always global.
Okay. And obviously, so the consortia got a hold of whatever experts they could in the respective countries, and to try and make sure that as many species as possible could be represented. Because I know just from our experience, I think sometimes, not all of the same can be said for GBIF. Not all of the species are on GBIF. So, I mean, obviously with the extension, if you do get the new funding, you'll cast the net as widely as possible. And it's always a work in progress. I mean, I found with FBIS, you're always updating the master list, especially for things like invertebrates, probably less so for fish.
Yeah, I mean, the idea really is, so what they did within FADA, I was not involved in this project, and it's a pity that Kuhn is not here, but because he could explain it more properly. But they collected the species per region, so Arctic region, Palearctic region, and so on, and not per country. And for many organism groups, there are already consortia. For example, for the caddisflies, there is the caddisflies world checklist, and also for Blackoptera, I know that there is a world list. So there are already consortia, and these consortia would be revived, and they we would just check that we really connect people who are not connected yet. And then the idea is also to put all this information into a curation system so that it can easily be updated. The problem with FADA was that it was just a kind of static web page. So if an author or if an expert found a new species, he had to ask the FADA technical person to put this into the list and so on. So my idea is either linking the FADA information to WORMS, so to the World Registry of Marine Species, because they are also hosting freshwater databases. So we could integrate these FADA databases right there, or we can integrate them into the TaxonWorks network. This is something, I don't know whether you know, but this is like the WORMS, but hosted in Illinois in America. And the third option is, of course, using your system, the FB system, and the taxonomy module of it to manage all these taxon information as well.
Okay, obviously I'm in favor of the third one, but
me too.
I think that would benefit everybody. So yeah, fingers crossed that we can generate some funding for that, because I think certainly, yeah, having a static system, I can imagine that would create problems, because you want the experts to be able to access the relevant groups themselves, and that's
Exactly.
when it's updated.
And then they can do it right when the new species appears or when there is a name change, and you don't have to ask them every half year, is there any change, because they are just doing it. So yeah.
And they have an interest, and so they haven't invested, they invested in it. So I could imagine. Thanks, Astrid. That's really useful. I'm quite keen to hear from Mo, if you can put your FBIS hat on, Mo, and just maybe talk us through the process that mostly you and Jeremy followed in terms of the generating the fish master list for the FBIS platform.
Sure, thanks, Helen. So I think our process largely focused on consultation with experts from around the country. So I was actually just working on that chapter of my PhD now, and we consulted close to 50 fish experts from around the country initially, and then our core team of researchers that ended up working on that master list was probably slimmed down to around 10 or so people. And I think that collaborative process meant that we worked through several iterations of our initial master list for the species in the country. And then from there, we went and conducted our data searches and found which species had data in the country and which didn't. And we ended up with a list of roughly, I think, 135 odd species in the country. There are about 7 or 8 species on that list where we did not find data, but experts from around the country were pretty certain that those species do occur in the country. And so our process, I think, didn't, because in South Africa, we don't have that many freshwater species relative to, say, for example, invertebrates, and it's quite easy to manage a list that's only 135 odd species. Our process worked directly with experts, and I think that's where a lot of our success was grounded in. In terms of updating the list, I think that's something that even now, as I'm kind of wrapping up the project, we're already finding problems with. So there are already 4 or 5 new species that have been published. There are an additional 20 or 30 lineages that are about to be described. And I think once those species become formalized, we will have the issue of how do we update our list firstly, but also how do we delineate our data. So one example that comes to mind is Pseudobarbus anapilus, which has just recently been split into 4 separate individual species. And on our platform, for example, that's all the same data. And so the process will have to include needing to update our list firstly, and then secondly, we would need to manually reassign data if that's possible. And it also brings up then an additional issue of, in the instance where a species co-occurs in the same area, or in the same geographic space, we're not sure how we would actually separate that data. So those are things that I think I'm already thinking about, and I know Jeremy is also kind of pondering. But for now, the system we have, because the freshwater fish community in South Africa is relatively small, we're able to, as Hugo has mentioned in the chat, we're able to deal with these kind of things as they come up and find solutions quite quickly. But obviously in a like broader sense and in terms of sustainability, I'm not sure how long that might be able to go on for without additional funding or some sort of automated system. And I kind of like the idea of allowing users to be able to update the list as they go ahead. And then lastly, to just talk a little bit about how our list relates to the GBIF backbone or taxonomic backbone. So where possible, we've tried to stick with the updated information that's on the GBIF platform. But obviously there are exceptions where these species have been updated in name or in lineage, splitting, for example, where GBIF is lagging behind in how quickly they're able to update their taxonomic list. And in those instances, we've had to kind of, on an individual basis, make decisions about which name do we go with, i.e. do we stick with the scientifically correct name that's recently been updated, or do we stick with the name on GBIF, which allows us to access that data? So that's also one of the issues we're facing now, which is for a few of our species, we're having to use an older name just because if we use the updated name, we're not able to pull down all of those occurrences. And that's something I'm not sure how we would go about resolving, because it involves having to get GBIF to update the name. And that, I assume, takes a few years, or takes a formal, like, I'm not exactly sure if anyone, maybe that could be a cool discussion point. If anyone knows how we can go about asking GBIF to change those names efficiently.
I'm assuming a lot of the fish data on GBIF comes from SIAB, so perhaps it's really the mandate. So SIAB is the South African Institute of Aquatic Biodiversity in South Africa, that they publish the fish data to GBIF on a fairly regular basis, or once or twice a year. So I would imagine the mandate for updating species would probably lie with them, because no one at GBIF is going to do it. It's going to have to be the upstream source, I guess, the information. Do you know, Astrid, what the process is at all for updating taxa on GBIF?
I have no idea how this works for the fishes. But I know for the invertebrates that we are, the process would be that we propose a list to GBIF, and they just decide whether they want to use it or not. The issue we are mainly having is that their system does not foresee a tagging whether a species is freshwater or not freshwater. This is not so relevant for the fishes, I admit, but we need this for the birds, for example, and yeah, also for parts of the invertebrates. And as long as they don't have this tagging system, yeah, we cannot submit our lists to them, or we do not want to submit our lists to them. So, but
yeah,
we see.
Sorry, just before you carry on, I totally relate to that, because when I first started generating the invertebrate taxonomic list for FBIS, my sort of first point of call was GBIF, but there was no way to just extract the freshwater species. So I realized quite early on that clearly wasn't work. And then what we did was, or what I did was to we have a really good set of aquatic invertebrate guides published by the Water Research Commission about 10 years ago. And fortuitously, I've been involved in a trace project a few years before, and we had a sort of a straw dog list of invertebrate species. And then we also approached Sanby and looked at, because they've got some checklists, although some of them are out of date, but really had to cobble together from, various sources, a master list, and it's continually changing, being updated all the time.
Yeah.
But yeah, I can relate to that issue with no tagging for freshwater.
The idea of this FADA 2.0 project is really to have the experts, and then as soon as we have the updated lists, make them taxonomic backbone of GBIF. And we have recently very good contacts to GBIF. We are doing a lot of other work for them as well. So we will write a best practice guide on how to publish freshwater biodiversity data on GBIF. So I'm fairly confident that we will end up with our lists on GBIF, but it's still a bit of a way to go. But if we are there, then these lists could be used by you as well, of course. And for sure, if we get in contact with GBIF, we would, or if we get to manage the money for FADA 2.0, we will certainly get in contact with you, so to see that these lists that we are producing really also contain all your species as well.
That would be great. Yeah, I think that's where this networking group that we've created is actually really useful, because we've got, the individuals who have been slugging away producing those lists, and like Mo, like you said, it's been a huge consultative process with a lot of input. I think the list started with over 200, and then it got whittled down with various insights and inputs. So yeah, it's a slow process, but once a master list or a checklist has been produced for a country, for a particular group, as new species are described, and like through FADA, I think it would be really valuable to have those experts like the Helen Jameses of the world for the mayflies, and I'm not sure what other experts there are, and that does bring us to the other thorny issue of the lack of taxonomic expertise within Africa, actually. I know a lot of Europeans and people from the States have done a lot of fantastic work in Africa, but I don't know to what extent that capacity has been brought down into the African institutions. It's probably a topic for another discussion, but just, without good taxonomy happening in country, the lists can, not mean an awful lot, I guess. We're lucky we've got Albert Chacona doing so much work on the fish, although he's making our life a headache too, because he keeps describing new species, hey Mo?
Yeah, I would agree with that. And as much as it is a headache, I think there's also a lot of work that has to get done that Albert is aware of and all of us are waiting for, but will take several years for that to get published. And I think there definitely seems to be a lack of capacity to actually describe those species. And in the situation with fish, because there aren't that many species, I mean, it's something that with enough funding and students on can be done quite quickly. But I assume with the inverts, the problem is probably much larger.
Yeah, but I have to say, in this global list, we have a variety of African or South African experts. So Ferdi de Moor for the Trichoptera, or Helen Barber James, who isn't in Southern Africa anymore, but they are, at least they know what is happening in your country. And there's, as you said, really much work going on from the Senckenberg Museum regarding taxonomy with this AfriTax stuff based mainly on molecular or on eDNA. So we would, of course, try to integrate all these different persons working in Africa. I don't think that the situation is too bad Yeah, at least in South Africa, compared to, I mean, other remote regions.
That's encouraging, except Helen's left the country and Ferdy's retired. So we need some young guns. We need some young Mo's and, yeah. But I think it's a challenge all over. I know that other colleagues in the series like Ciro and the Rwandan team that I've been working with, they also feel quite hamstrung in some respect, by the lack of taxonomic expertise within country. But yeah, it's
yeah, but this is certainly not something that is only limited to Africa. I mean, these taxonomists they are dying out. So in Europe, they are now establishing a red list of invertebrate taxonomists, just to see how much expertise we still have in Europe. So this is something really that is done by the European Commission with the help of the IUCN, of course. Really to see where do we need more experts, for which invertebrate groups we would need more experts, and hopefully this then will again lead to that taxonomy is something that gets funded. Because the problem here in Europe is that this is a job, you don't find a job. Yeah, if you want to do taxonomy, you can do it in your leisure time, but it cannot be the job that feeds you. So yeah, this is something we also have to fight with.
I hear you for sure. And you'd think with the eDNA being sort of very much pushed at the moment, that it would have sort of spring-started a little bit of extra taxonomic focus, because you can't have eDNA barcoding happening unless you're actually confident about the species.
Exactly. And I was attending a conference earlier this week, and someone from the European Commission, so a very high person, he really also said this. We need taxonomists, otherwise we cannot handle all these molecular data if we don't have the real old morphological taxonomists. So if this is already recognized in the commission, it might be a way forward.
Yeah, that's encouraging that it's at least been recognized as a hindrance, and so with luck something will happen. Hugo, I see you made a comment, but I think Mo probably spoke to that essentially.
Yeah, no, he did. Sorry, I don't know, when I'm on the Mac, I can't find the icon for the hand signal. So, I mean, I think Mo definitely addressed that. And I mean, I'm not really a taxonomist. Well, I'm not a taxonomist. So I'm not going to say really a taxonomist. I mean, obviously my focus is more, on modeling and the abiotic factors that drive the biota ultimately in the system. But, Mo mentioned how the, one species of Barbarus is now becoming 4. And, just from a kind of technical perspective of the data platform and, coding in to account for that, because obviously any species previously would have been logged, as a clumped, before it got split. So it'll be interesting to see how you deal with it. And I know that there are not many species, obviously, of fish compared to, say, the macroinverts. But like my partner, Meg, she did a master's and she was looking at the river sardines with Prof. Skelton and them. And she was able to actually identify 2 new species and name them. And it was records of samples that had been taken like 80 years ago that were assumed to be the same species. So, I mean, more and more of these genetic studies are being done. So I think things are going to become quite hectic in the future, because if you're getting master studies pulling out new species, I mean, it's definitely going to cause havoc with databases and platforms like FBIS, for example. So it's just interesting hearing, like, the concerns already raised and, what kind of approaches you'll take.
Yeah, I mean, we've kind of spoken around it quite a bit, but we certainly don't have all the answers yet. I think the best case scenario is that a new species is regionally distinct, and it's the only species. So, some of those lineages that only occur in a specific area, I guess one could make the assumption that, any species that was called species X, that's now species XX, gets reassigned to that species. But yeah, as Mo indicated, the problem arises when you've got more than one sort of new, you've got two species that have come out of one and they cohabit the same area. I don't think there's going to be any way really, unless the samples are still in existence, you know, and they can do eDNA testing on them. But a lot of the data in FBIS doesn't have voucher specimens associated with it. So we would just need to, I guess, leave it as the original species name and then have a sort of a date at which the species changed. But yeah, it definitely represents quite a big challenge for managing data.
Because I can imagine, I mean, especially like if you take the case of those river sardines, the only way you were going to be able to tell that they're different species is on the genetic level. Now, I don't think any person who's going out and sampling fish are ever going to go to, you know, the genetic level every time. So, I mean, it has to be defined by a region if possible, if they're obviously not cohabiting the same space. And in her case, luckily, it was 2 river basins and she was able to show that they were separated or something of that sort. It was part of her study. But yeah, I still think it throws a spanner in the works for you guys, and it's a big challenge to address it.
And obviously just not for the sake of, you know, calling a new species, but it has repercussions for red listings and, you know, conservation status and what have you, in terms of like endemism and what have you. And can, you know, if you think of it in terms of like how we're trying to integrate FBIS into the decision tool frameworks going forward, like the EIAs, you know, a highly threatened species will trigger a much bigger response than, you know, least concerned species. And so if you've got a new species described that's got a much more restricted range, that would trigger a more severe response. So yeah, there are some long-term sort of decision repercussions or management repercussions interlinked with the taxonomy.
That's interesting. It's almost like, you know, identifying these levers of change. So like potentially utilizing the species to really change the course of development in areas.
Exactly. Mo, your hand's up, go for it.
Yeah, thanks, Helen. I think to add to both yours and Hugo's points, so for some of the species, actually, like separating out the data has not been a difficult task. And so in terms of the EIA policy and the screening tool that's come out of that, we were able to delineate, I think 7 or 8 lineages within the country that were previously clumped together. And the process that we had used for that was to contact species-specific experts. And we sat on a Zoom call with a GIS screen up and went record by record and asked them to separate the species out for us. And whilst that process was tedious, it actually worked really well, where we were able to generate these distribution maps for species that haven't yet been described. And so for those data, for example, as soon as the formal description comes out, we'll be able to separate that out. And I think that will be hugely beneficial, especially in the like EIA context. But I think for other species, for example, one that pops into my head is the Galaxia suite that's going to be separated. I'm not sure how we would even start to think about delineating that. And it would possibly require like a couple of PhD students who become experts on those species to be able to sit and like truly provide a delineation of where the data might lie. But yeah, I think the process will probably end up being something along the lines of, for most of the lineages, we'll be able to do that separation with the experts coming on board. But for some of them, it's going to be near impossible. And yeah, I think that's unfortunately just something that we have to live with.
Yeah, no, I hear you. Good, thank you for that. So I don't know, unfortunately, we haven't got any of the biodiversity focused people in the room, but I was hoping to get a sense of if any of the projects, the GRS funded projects, have used the FADA lists for assisting with the generation of the lists. But yeah, I guess that's not. Yeah.
How was it done? How was it done for Rwanda, for example? So in the countries where you use the FBIS, how was this extended then, or how did you do it with the taxa list?
So I mean, I trained the team in how to generate the lists, you know, and I know for the fish, I think their first protocol was actually fish base initially. And then there was a published checklist of fish in Rwanda. And I think they used the combination of that. I don't think they actually did go to FADA, but it would be quite interesting and that was one of the reasons I was trying to download some of the lists. I wanted to do a sort of a check between what the FBIS master list was compared to some of the groups on the FADA to see to what extent we'd either missed species or, you know, hadn't included everything. And it would be nice to try and encourage the team from Rwanda to perhaps do the same. Just as a check, you know, to see to what extent Africa is covered. Obviously, I know for some groups it's done really well, like the ones you mentioned, the Plecopterans, the Ephemeropterans, and I think the fish species are also pretty well done. I mean, I'm not sure. I haven't looked extensively at FADA.
I think the fish list really comes from FishBase or from the FishBase consortium.
FishBase,
so.
yeah. So I guess, and I know with the algal database, there's also algae base. So okay, so FADA does already link with some of those other well-established databases. Is that how I understand it? Yeah.
I just want to check, Helen, when we refer to GBIF and FishBase, I know FishBase is integrated into GBIF, if I'm not mistaken, so you can access directly through GBIF. So when we refer to FishBase, we're not referring to it as an individual data set, but rather a subset of GBIF connections or, you know, API connection. I'm not too sure how it exactly works.
Yeah, I'm not sure. Is that a question or a statement? Because I don't know.
Well, I'm not too sure if it's a question or a statement, but I know that, you know, FishBase you can access independently. But then also FishBase has a component within GBIF. And I'm not too sure when people say, okay, we're going to be accessing through GBIF, that they're also then accessing via GBIF FishBase, if that makes sense.
Yeah, I can't really answer that. I think it is all integrated in the back end. But I know that we've been working quite a lot on the algal master list because, you know, for phase 1 of FBIS, we generated a master list for algae that were in river systems, and now with expanding to inland wetlands as well, obviously we've got a whole lot of new species. And when we looked at the GBIF versus the algae base, there were a lot of species that were in algae base that weren't in GBIF. So I don't think it does connect in the back end via an API. I think the two are separate, but I stand to correction. And then the other issue was even higher up, even like the taxonomic classification differed between what was on GBIF and algae base, and sometimes quite high up, you know, at sort of family and even higher. So I think, yeah, I don't think it's a very clear system, and like that's why, I mean, Tim, you could jump in here maybe as well. You know, at the beginning of the FBIS project, we took the decision to use GBIF as the taxonomic backbone, knowing that it wasn't perfect, but that it was kind of the best available at the moment.
Yeah, and I mean, it's not my domain expertise to know what the best is, so I mean, I sort of rely on you guys to tell us. But I do think it would be, I sort of said in the chat there, it would be good to maybe try and get the GBIF folks into a call and tell them what our issues are. And, you know, we've got other things to discuss with them as well about publishing a current state and so on. And maybe there's a lot of stuff we don't know about what they're doing in the background and so on, to sort of get the inside track from them.
I think that would be really useful, especially Astrid, when we have that discussion around, or follow-on discussion around that, improving the taxonomic management side. I mean, the system we've got in FBIS works for now, but it's by no means perfect. And obviously it's being deployed in other instances. So if we can, find some additional funding to improve that, and then definitely have a conversation with some of your connections at GBIF to really get an inside perspective, because it clearly is something that is a help or a hindrance, and it can be one or the other or both, depending on which taxonomic group you're working on, and then how that then links with the more recent FADA. So Hugo, you chirping in the chat there, you saying FADA?
No, sorry. I'm just
GBIF.
Googling some of the links. I mean, it's interesting. I mean, it seems like GBIF is essentially either, through API links to all the different databases, but it seems to be a central point where you can access all the data sets. And I think it's more just important for us always to remember, if we're going to reference any data sets or data that's coming from somewhere, that we don't just say GBIF, but maybe actually look into where the data is coming from, further on.
Definitely. We've got that in our CSV file, so that when the user downloads the data, you can actually drill down, oh, it's the SciAb database that's been uploaded, or it's our naturalist research grade data, etc. So that is specified. But I hear you, that's really important.
But regarding your last comment, Hugo, the FADA lists are parts of the FADA lists are accessible through GBIF, meaning that we have published them with GBIF, but they are not using them as taxonomic backbone. This is just an information document, but it is not the backbone of GBIF.
Okay, thanks. I remember you telling me that before.
I just saw the download core Darwin archive and just assumed. Sorry, I didn't realize that it wasn't the backbone.
So the interesting thing about GBIF, and I'm definitely not the world's expert or even the FRC expert on GBIF, but you get the taxonomic backbone, which is kind of what we use. That's with like a little separate room in GBIF, and then you've got a whole lot of other information sources that are not necessarily used in the taxonomic backbone. That's the way I understand it. Hey, Astrid? Yeah, okay. And I think FADA is one of them. So it is in GBIF, so it's accessible in GBIF, but then they haven't yet integrated it into the taxonomic backbone. And that, I guess, is the focus of your future project, to try and get that integrated, which I think will be excellent. Okay. Good. So we've kind of spoken to the first three of our questions, and I guess the next thing is sort of how can we as a group not only obtain, because that's part of each project's workflow, but create up-to-date taxon lists going forward. And Astrid, as you mentioned, there are plans afoot to see if we can up the value of the taxonomic management system within the FBIS or the BIMS suite of projects, and use that in some way that allows local taxonomic experts or international taxonomic consortia to each update their own species on the central system. And I for one think that will be a really good way forward. I mean, Mo, you can also imagine the likes of Albert, as he publishes a paper on a new fish species that's described, and he has access to that taxonomic management module, that he can then publish the paper. Once it's published, go up and update the taxonomy. It won't necessarily drill down to the actual data. That will still require some sort of administrative intervention depending on the regionalization of a new species. But I think that's probably quite a useful way going forward. It'd be good to just get some general thoughts from everybody.
we have been recently in contact with both of these taxon management systems that exist basically out there, because we want to move the Trichoptera world checklist to one of these two systems. So the one is the AFIA system that is integrated with worms, and the other one is this taxon work system from the university in Illinois. And basically both work the same way. You are, for example, an expert, a fish expert of a specific country, and then you have an expert a level above this, an expert from Europe, for example, and then you have a final committee for the world checklist. And the idea is, so if I'm an expert in my country and I find a new species, then this will be validated by the next level. And then when the editor of the whole group says, okay, this is okay, he can just tick a checkbox, and the species would go from the lowest level online, so to say. So there are different levels of access, what I think is very useful, because then you can also have your country experts in the group, but you have still someone who is managing the whole group. It's really a community effort then, rather than one person managing a whole checklist for the world.
That sounds really, really good. I'm excited at the prospect of that happening. Yeah. So and also having the different levels in terms of quality, control and just validation, I think is really important. Excellent. I think we've lost a few folk. So Mo, did you get the details, the link to the FADA, the list, and you can integrate, you can query it and what have you. I know there were a few broken links, but it seems to be working now. I did dive in just before the meeting.
Yeah, I just got it now. Thanks, Helen.
Okay,
I'm going to spend some time playing around with it this evening.
It would be quite interesting to see how that compares with our master list, just for a specific country, just as some insight, give us insight into the extent to which they match or they mismatch. I know we've even had big mismatches with the National Biodiversity Assessment checklist, because sometimes the criteria differ, in terms of either defining what a freshwater species is, or in terms of the extent and what have you.
This information would really, really also be very important for our FADA 2.0 proposal, so that we really know. So maybe Mo, if you can do this check within the next 2 weeks or something, and then let me know how much difference there is, because then I could also add this to the proposal to underline the urgency that we have an update actually of these lists. This would really, really be a valuable help in moving these things forward.
That's great.
Sure. I can get your details from Helen and send you an email once I've downloaded the data and seen what that list looks like.
That would be really cool. Thank you.
That would be great, Mo. And maybe what I can even do is ask my colleagues in Rwanda to do the same for their fish list. So we'd have it for 2 different countries, maybe one slightly better resource and one less resource, that would then just provide more motivation, I guess. Fantastic. Thanks, Astrid. Yeah, that'd be great. Always keen to add ammunition to the proposal in whatever way we can. Excellent. I'm happy with our chat. It hasn't been a big group, but for me, I've learned a lot, and I certainly think we've made some good connections. And Mo, I appreciate your insight, because you're coming from a place of having literally been at the cold face and having done it yourself, so you know exactly what it's like. Mike, any insight from your side? You've been very quiet today. I know you're also not a taxonomist, but you've always got good insight.
Yeah, for me, it's mainly interesting to see how things have progressed. Way back when Jackie King and I worked on the wetlands, we had a small GBIF3 database, and I was just looking on the computer now. We actually had it was in a Botanical Research Institute, they had a list of codes for various species. So it's something that people have been working on for quite a while. What always catches one out is when the system is too rigid and you can't cope with some splitting and lumping of species and things like that. And then you also mentioned the actual loss of taxonomists, or maybe the downgrading of taxonomy as a profession, but it's really important.
Agreed. Yeah, really critical. I know that we're about to publish the plant module onto FBIS, the wetland plant module. It's going to be the next one. And the SANBI team have been working furiously to develop the plant master list. And they started out with 948 accepted species and then wanted to include all the synonyms, because a lot of people are still using synonyms for capturing data, or the accepted species on GBIF or the synonym, it's reflected as a synonym on GBIF, but in other databases that South Africa uses, it's accepted. So there's now over 2,500 species, and 1,500 are supposedly synonyms. So it becomes such a nightmare. It really is. I mean, you look at one plant species and it's got 20 other names. It's completely and utterly crazy. But yeah, I'm glad I work on bugs. And Mo, you can be glad you work on fish. Yeah. Okay, so thanks everyone. Unless there's anything else anyone wants to add, anyone for a last chirp, or we can.
One last chirp. I'd just like to say thanks, Helen, and Jem isn't here, but I mean, the seminar series have been absolutely fantastic, and I really appreciate all the time and effort that went into them. And yeah, they've been really, really valuable. Thanks.
Great. Thanks for that. Yeah, sadly, this is the last one for now. We're not sure what will happen going forward, but for 2022, at least, this is the last formal seminar series. But we've made some great contacts across the globe and across the continent. So I have no doubt we will all be connected and remain connected going forward. What I personally would love to see is to try and get a conference funded, that brings everybody. The seminar series started in the form of a workshop, which, as we know, didn't happen because of COVID. And I think a lot of the projects and the team members have continued with their work or gone on to doing, slightly new things. But there's a lot of useful information and shared insights out there. And it would be really fun in a way to maybe organize a bioinformatics, freshwater bioinformatics conference, but maybe have it in Africa, but not limited to African representatives. So maybe that can be something we can strive towards in 2023 or the year after.
That would be really cool.
Okay, great. Maybe we can hatch up a plan then, Astrid, and see where we can.
See that we get a bit of funding for that.
Exactly. And yeah, I think it'll be great. And I think it'll be wonderful to meet all in person, and it wouldn't just be exclusively this group, but, cast the net much wider and just bring together a north, south, east, west group of people that have all got similar interests and, shared values. Great. Excellent. So thanks again, everybody. And yeah, stay well, and we'll connect independently going forward.
Thanks, Helen, and all.
Great. Thanks so much.
Thank
Bye-bye.
you.
Everyone.
Cheers, Mike. Cheers, Mo.